All right. Hello, everybody, and welcome to the reintroduction of the Green Bank Observatory Colloquium series. For right now, this is going to be very familiar to all of you as our science lunch talks, but we hope to, in the future, branch out, possibly moving this to a different time in the day and or week, while also, for those of you who will eventually come on site, reintroduce the in-person science lunch talks. So today I have the pleasure of welcoming Ceci Zhu, a PhD candidate and Reber Fellow at the University of Virginia. And she will be talking with us about the latest results of the Gotham survey and interstellar cloud chemistry. With that introduction, I want to remind everybody, please remain muted during the talk, but there will be a question and answer period at the end of the talk, where you are welcome to unmute yourselves or submit questions in the chat. So yeah, with that, take it away.
Yep. Good afternoon, everyone. Thanks for the introduction and having me for the GBO Colloquium this week. And thank all of you for tuning in. My name is Ceci, and I'm a graduate student at the University of Virginia. I have visited the observatory by this time around 2 years ago for the Gotham team meeting. And I still remember how impressive the autumn landscape I saw along the US world, I think it's 250. Yeah, so hope I can have a chance to get back there in person in the near future. Okay, so let me get back to my talk. Today, I'm going to present my graduate work on molecular relationship in interstellar molecular cloud. Actually, I'm going to have my thesis defense with this similar context next month. So any suggestion or comments is extremely welcome. Here is the outline of today's talk. The talk will be divided into two parts. First, I will introduce the molecular inventory and in the interstellar medium and the contribution from the GBT as well as the recent Gotham program to the molecular detection. Secondly, with two examples, I will then discuss in detail how molecular spatial distribution help us to investigate both the chemical relationships and source physical structures. And these studies are targeting the interstellar molecular cloud, ranging from dark cloud TMC1 and whole molecular core stage B2 and NGC 6334I. Let's dive right into the molecular sensors. Starting with some numbers, until now, we have identified around 240 molecular species in interstellar medium, among which the single dish telescope have contributed to more than half of the new molecular detection. Here is a nice plot from a census paper published by Dr. McGuire, summarizing the facility contribute with 10 or more total detection. They are all single dish telescope operating at the radio wavelength region. And GBT has contributed almost 30 new species. There are lots of fantastic large observational programs performing with GBT, and I'm lucky enough to be involved in the Gotham program as a co-PI. It is a deep chemical survey with around 850-hour observational time directed toward the TMC1. TMC1 is a filamentary dark cloud located in the Taurus molecular cloud. It is the closest molecular cloud we are going to talk about today, with astonishing chemical richness. Of the more than 200 interstellar molecules, around 60 species were discovered toward this source. In addition to expanding the molecular inventory as the initial stage of star formation and a representative of dark cloud, TMC1 provides the initial condition for chemical models of later phase. Assisted by the joint effort of the deep observations and the detection techniques, our team has reported the first astronomical detection of 12 new complex species toward this cloud. You might have heard about the talk several weeks ago presented by one of my colleagues, Mark Sieber, about the detection of methyl carbon chain. Here is another example molecule, HC4NC. The discovery was achieved by the identification of 3 emission features. The observations are showing black, while the simulation are showing color. Studies of chemical composition in this source have provided us the information about molecular abundance and their excitation conditions. The observational information synthetic with the astrochemical modeling further enable us to investigate the chemistry of the molecule. For example, by comparing the observed and model abundance of all the isocyanide and their counterpart cyanide species, we can further constrain their relation pathway and the chemical model under dark cloud conditions. Another example is the cyanopolyene chemistry presented in Loomis et al. Published this year. For a long time, TMC1 had been assumed to be homogeneous dark cloud with an extended morphology. However, as shown in the spectrum, benefited from the high spectral resolution of Gotham program at about 0.01 kilometer per second, we are able to resolve 4 cloud components with distinct systematic velocity. To synthesize the spectrum of one component, we need to assume 5 parameters: the source velocity, the source size, and the column density of the molecule, the excited temperature, and the line width. So for 4 components, there are 20 parameters in total to fit. This is very large space, so we reduce the number of parameters by assuming uniform excited temperature and line width. Therefore, what's left are 14 free parameters. Also, the Monte Carlo Markov chain analysis help us constrain the information. If you take a careful look, you will notice the large uncertainty in abundance and the source size. In the following, I will explain the source of these uncertainties. Here is an example corner plot of the MCMC fitting result of another molecule, HC9N. The corner plot shows the parameters covariance and their distribution. For example, the first column shows the probability density function of the source size number 1, as well as its correlation with the rest 13 parameters. Let's take component number 1 and a few other parameters for illustration. The probability density function shows where the best fit parameters lie. And one way to read this figure is that the more elongated and tight the distribution is, the more dependent or degenerate between the 2 parameters are. For example, the top plot shows that there are weak covariance between the source size of component number 2 and number 1. However, something catch your eye immediately is this elongated distribution, which suggests strong correlation between the column density and the source size for this component. The pull constraint on this source size lead to a large uncertainty in the total column density. In addition, in this chemically rich environment, where cell self-extinction can play a role. The extent to which cloud component overlap strongly correlated with the column density. As demonstrated in this plot, the thickness of the dark gray line represents the intensity at the location, considering both contribution from molecular emission and self-extinction. If the four velocity components are spatially overlapped on the co-spatial regime, the component from the more distinct, the emission from the more distinct component will be further attenuated. The self-extinction and thus the observed intensity are different from what we can get under the spatial separated regime. This observation showed that the lack of spatial information poses a problem in estimating the abundance of molecules and the chemical investigation. The second part of today's talk is exploring the molecular spatial distribution with two cases of study, including the comparison between the morphological difference of isomeric family and that of a specific molecule, but in different energy level. This study was achieved with high spatial resolution interferometric observation, namely ALMA. The first case is the astronomical triplet. So triplet, are we going to talk about babies? No, they are the molecular triplet. The molecular triplet is a family of certain kinds of molecule. Chemists usually call this kind of family as isomer. They have the same number and the same type of atom, but connect in a different order. Isomer can act as tracer to elucidate temperature and density-dependent formation route to synthesize complex interstellar molecules. In addition, further insight can be gained into the astronomical evolution of different physical components of the source by this isomer. Now we are focusing on the isomer that consists of 2 carbon, 2 oxygen, and 4 hydrogen atoms. They are methylbromide, glycoaldehyde, and acetic acid. Methylbromide is ubiquitous in molecular clouds, and its formation is suggested to involve both gas phase and green surface reaction. Glycoaldehyde was first detected with the 12-meter single-dish telescope, and its extended distribution suggested with the BIMA array observation indicate gas phase formation mechanism for it. Acetic acid, a component of the vinegar, has only been detected with interferometries and always possess a warm, compact distribution, which favors green surface formation mechanism. CB2 is a giant molecular cloud located near the galactic center and the most massive star-forming region. CB2 is also famous for its chemical richness. Many interstellar molecules were first detected toward this region. In fact, CB2 is the source that contributes the highest number of molecular detection. We have talked about TMC1 dark cloud before, and TMC1 is actually the second place. The spectrum towards CB2 is like a line forest. There are more than 10 lines every 100 megahertz, and it's very hard to find any line-free channel in this spectrum. The large line width and the high density of lines makes majority of the line of these 3 molecules is contaminated with the emission of other molecules. Let's take glycoaldehyde as an example. As shown here, it is a summary of some of the previous detected glycoaldehyde transition towards HB2. Almost all of them are blended with other molecules. A widely spread opinion about glycoaldehyde is its diffuse spatial distribution. And if you check their reference, the only available map of glycoaldehyde towards HB is from the Hollis et al. 2001 with the Beamer observation. However, in their research, the spatial distribution of glycoaldehyde is based on only one transition, and it is this one. The broad line width of the line, which is around 24 kilometers per second, compared with the 8 kilometers per second for the methylformate line in the same observation, highly suggests that it is a contaminated transition. They suggest the source size of glycoaldehyde is around 1 arc-minute with only this transition. So the conclusion of a diffuse distribution of glycoaldehyde towards the edge is most likely inaccurate based on this observation. This plot shows the spectrum of some previous reported transition of glycoaldehyde covering our frequency range. The black line represents the observed spectrum, with while the glycoaldehyde is simulated in red. As you can see, they are highly blended and only a few are clean. In contrast, this plot shows the spectrum of the 10 most uncontaminated transition of glycoaldehyde that we found. The uncontaminated transition enable us to image molecular spatial distribution properly. This plot shows the peak intensity image generated with the least contaminated transitions of glycoaldehyde, judging by the spectrum at one position. You can notice on the top of the spectrum contamination, we still need to deal with spatial contamination in order to properly characterize their morphology. These three plots show significant different distribution from the other, as we should expect the distribution of similar energy level of a molecule should behave similar. So most likely the difference is caused by spatial contamination from other molecules. Note that this information is only accessible when we examine transition maps of multiple transitions of a molecule. Using a map based on one transition to represent the morphology of a molecule is like computing the means of one sample point. The sample variance is infinity. Other than the contamination mentioned, the remain peak intensity image of glycoaldehyde are consistent with each other. Here are the less contaminated transition of methylformate and its transition maps. Despite the spatial contamination, different transition maps of methylformate show nearly identical spatial distribution to each other. By averaging the uncontaminated transition maps weighted by their noise level, we have the so-called chemical map of the three molecules. Compared with mapping a single and maybe potentially contaminated transition, this gives a much more robust way of mapping molecular distribution. The three maps show the spatial distribution of methylformate, glycoaldehyde, and acetic acid respectively. The background is the continuous emission, whereas the control represents the molecular emission. The distribution of methylformate is extended and offset from the continuous emission, whereas the overall distribution of glycoaldehyde and acetic acid are more compact and co-spatial with the continual emission. What does it tell us about the chemistry? Here, the diagram summarizes the prevalent formation mechanism relative to the 3 isomers and other species. Let's take the most popular radical-radical reaction on grain surface, for example. Glycoaldehyde and methylformate have the same precursor HCO radicals. In contrast, acetic acid is formed from a totally different precursor, the CH3CO radicals, which is called secondary radicals in Garros et al. 2008. So if this scenario is the case, it is reasonable to suspect that glycoaldehyde and methylformate have similar observed behavior, but different from acetic acid. However, what we find is that glycoaldehyde is actually have similar spatial distribution with acetic acid. Another suggested scenario is that acetic acid and glycoaldehyde are formed on the grain surface through those radical-radical reactions, whereas methylformate is formed in the gas phase. The distinct spatial distribution of methylformate and the other 2 isomers give us an insight their relative role of gas phase and grain process in forming them. The last part of today's talk is the distribution of acetic acid in different energy levels. For the same molecule species, emission region of high energy transition are not always identical to that of the low energy transition. A few studies work on spatial mapping the transition of a molecule covering a large range of energy levels indicates that complex molecule may uncover underlying substructure of the source. The third molecular cloud of today is a massive star-forming cloud, NGC 6334I. The NGC 6334 region is also known as Keppel Nebula due to its similar shape to the Keppel. It contains multiple signs of high mass star-forming region. The deep embedded source I is at the northeast end of the region and has been reviewed with rich chemistry. The ALMA observation further resolved the region into 4 main sources: the ultra-compact region Mn3, that's called Mn4, and chemically rich hole called Mn1 and Mn2. We perform a dedicated identification of both first and second vibrational excited state of acetic acid, and this project actually reports the first interstellar detection of this molecule in vibrationally excited states. The highest energy transition we detect has an up-level energy more than 300 Kelvin. Again, here are the cleanest transitions showing red and blue for the first and second vibrational states respectively. Here are the peak intensity mass of vibrationally excited acetic acid overlay with the continual emission in CON2. The 2 maps at the bottom right were removed from the following analysis because of spatial inconsistency and possible contamination. As in the remaining plot, we resolve 3 prominent emission peaks of the molecule marked as triangle, circle, and square respectively. 2 of them are within the Mn1, whereas the other is located toward the Mn2. The emission peaks are further pinpointing with the substructure of the region. 7 dust clumps will resolve toward the hot core MM1, namely A to G. One acetic acid emission peak is associated with the densest core MM1A. Another shows a dispersed distribution. They lie between the hottest MM1B and MM1D compact component. The variation of the position of the molecular peak is consistent with the large scale outflow structure in the northeast direction, originating from the MM1B. The hot core MM2, however, exhibit a core halo structure. The acetic acid emission peak associated with the MM2 clustered into 2 groups depending on their energy level, which are separated by around 0.3 arcsecond. And the emission peaks pertaining to the lower excited state are located toward the MM2 core region, whereas the higher energy transitions peaks are located in the halo region toward the southwest. This offset could be explained in part by the dust opacity. In addition, the MM2 is actually projected onto the large-scale blue sheaf flow of the MM1 outflow. Therefore, 2 mechanisms invoking dust opacity and outflow are both possible to lead to the correlation between molecular peak position and their up-level energies. Here is the summary. Let me just go over the result. First, the Gaussian program has contributed to 12 new species to the Interstellar molecular inventory. The covariance of molecular abundance and their spatial distribution make it clear that we must consider the spatial dimension when studying the molecular relationships. The high-resolution ALMA observations uncover the distinct morphologies of the C2H4 two isomers, which suggests that methylformate has a distinct formation mechanism from the other 2 isomers under the physical condition in ZB2 North. And the dependence of emission peak position of acetic acid over a large range of energy level brought to light the potential unresolved substructure and outflow of the NGC 6334I. And it is the end of my talk. Here is the acknowledging information, particularly thanks to all my co-workers. And thank you for your attention. I'm open to any question.
All right. Thank you very much, Ceci. That was a beautiful talk.
Thank you.
Yeah, so as you stated, everyone is free to unmute yourself if you wish to ask a question. And you're also welcome to write any questions you have in the chat, and I can read them out loud.
I'll ask one. Thank you for a good talk. I might have missed it, but when you're showing those corner plots, why was there so much covariance between the column density and only one of the components, but not the others?
So in this plot, I only show the component 1 because I can zoom in that, it's larger. But if you go back to the complete corner plot here, you can see this is the component 1 with the column density of the component 1, and that's for component 2, 3, and 4.
Okay, yeah. So now I understand. You're showing the column density from each different component separately. Okay, yeah, that makes sense.
Thank you for asking.
All right, Tony, you have your hand up.
Sure, this should be an easy question. But when you showed the spectrum of the galactic center there with the line force that you described, how do you actually select a continuum to continuum? Where's because what you did show later was a SAGB2 continuum in grayscale, and then the emission from the molecules on top of it in contours. So how do you go about selecting the continuum for such a difficult and line forced kind of spectrum?
Actually, it is not the easy question. Actually, this is the hardest part in my project to identify the light-free channel. However, thanks for the help from Dr. Thor Hunter, he designed an algorithm that combines the spectrum not only toward one specific region, but also different regions within that source, and take an average of that spectrum, but from that spectrum to design the light-free channel.
All right. And so are there any other questions from the audience? Yes, in the meantime, I had a question just looking at the isomer triplets. So you were able to at least get a sense of the different formation mechanisms that separate, I believe it was the, if I can remember any of their names, the acetic acid from the glycol.
Aldehyde. Mm-hmm.
Right. Are there any methods that you are considering to follow up to verify that information?
Yeah, actually, there are a lot of different mechanisms of both chemically or physically may contribute to the difference. For example, the desorption behavior may also be another explanation on top of the chemistry. So I think a follow-up investigation is that particularly recent, Dr. McGuire have a large program that is talking particularly toward these three molecules, but toward a large sample of different hot core. So because this spatial difference, what we currently found is towards GBO2, the only one source. If we can expand our sample point to more different cloud with different physical condition and the chemical composition of those sources, I think that's a very good strategy to verify which mechanism is the true reason.
Mm-hmm. Have there been maps of the dust grains in the source?
The map of towards edge.
Yeah.
As to my knowledge, I didn't know about the maps of dust toward this region, but there may be.
Okay, thanks. All right. So I'll turn it over. One last opportunity. If anyone has a question that they would like to ask.
I hope I didn't speak too fast because I noticed I'm kind of shorter than 30 minutes, so I may have to slow down next time.
You're all good. That's all right though. If you guys do have more questions, you can always get in contact with Ceci.
Yeah.
And so I will once again thank you very much for joining us today, and good luck with your defense.
Thank you. Thank all.
Hope everything is going well. And yeah, thank you everybody as well for joining us. Next week, we will have a returning guest in Natalie.